Good day, everyone. I'm Samuel Akigbe, a principal medical lab scientist with Maitama District Hospital Laboratory Unit. Today, I'll be taking you on Module 3 in the Diagnostic Antimicrobial Stewardship program. And we'll be talking on sample processing and identification with focus on the blood culture. Our outline for today includes the sample reception in the laboratory, rejection criteria, quality determination, incubation of the blood culture bottles, what to do if there is a positive signal, staining, differential media used in the laboratory, isolation of bacteria in the laboratory with emphasis on single colony, then trying to differentiate between pathogens and contaminants. Sample reception in the laboratory is based on certain known criteria, and when all these criteria are met, the sample will be accepted in the laboratory. In the case where these criteria are not met, there will be outright rejection of sample. And most of the reasons for sample rejection include incorrect container or transport medium, broken containers, poor sealing or leakages, unlabeled specimen, delay between specimen collection and arrival at the laboratory, incorrect storage condition, for example in the fridge, or greater than 40 degrees centigrade. It is pertinent to know that for blood culture bottles, if they cannot be analyzed or placed in the bat alert machine in the automated system as soon as they are received in the lab, they should be kept in the incubator, which is maintained at 36 degrees centigrade plus or minus 1 degree centigrade. And finally, inadequate specimen quantity. There are different kinds of blood culture mediums, such as the TBI broth medium, the biphasic medium, which has a solid phase and a liquid phase for isolation of pathogens, and also the automated systems, of which the bat alert is one of them, bat alert bottles, which is one of them. The bat alert system is an automated system of analyzing blood culture bottles in the laboratory, and it has so many advantages to it. And one of them is the colorimetry detection of microorganisms. And all this happen when CO2 gas is produced, when carbon dioxide is produced by the bacteria growing in the medium, and of course the sensor at the bottom of the bottle will detect those growths. All these reactions result in a color change, which is measured by the system. The second is a continuous monitoring of growth in the medium, which occurs every 10 minutes. And that's why it is possible to receive a preliminary result or a signal within 1 hour of incubation of the bat alert bottles. The third reason is flexibility, because it can detect both bacteria and fungi. It can be used for samples from other sterile sites, such as CSF samples, aspirate, and so on. The fourth advantage is adsorption of antibodies, and this is made possible by the presence of resins in the bottles, in the bat alert bottles, which absorb any antibiotics that the patient must have been on prior to sampling of the patient for blood culture analysis. So when this happens, the outcome of the analysis is not affected by the antibiotic therapy the patient has started. And finally, the unloading of samples. It is so easy to unload both positive samples when they have signaled, or at the end of the duration of incubation, the removal of negative bottles. The sensitivity of the bat alert system cannot be overemphasized, because it's a continuously monitored system. And at every time there is a signal, that sample can easily be attended to and analyzed with all urgency. And for the sensitivity has been seen to be increased when pairs of analyses are carried out. Apart from the pediatric, which has a single bottle, for the adult analysis, they have both the anaerobic and aerobic bottles, and collecting more than one pair has resulted in increased sensitivity drastically. It has been studies that show that collecting one pair of bottles has a sensitivity of 73.1%. Collecting 2 pairs of bottles, 2 pairs, has a sensitivity of 89.7%, while collecting 3 pairs increased the sensitivity drastically to 98.3%. In each bottle, at least 10 ml of blood should be collected. It can be a little bit less, but not more, especially for the pediatrics bottles. Also, when you talk about the incubation time, for any positive blood culture system, by day 1, it can be detected because the shortening of the incubation time and the sensitivity of the system. Studies have shown that by day 1, 69% of positive blood culture bottles can be identified and processed, and by day 2, 26%, day 3, 3%, day 4 and day 5, 1% each. Of course, in the laboratory or in the context of the country, of the Nigeria country, most laboratories might not have access to constant power supply running for 24 hours. And in that case, most of these analyses have to be done manually. In that case, all that is needed is an incubator, and the bottles that are placed in the incubator are plus 36 degrees centigrade, plus or minus 1 degree centigrade, and they are examined daily for growth by visual check. And this could be in the form of pellicle formation on the surface of the liquid, of the broth, gas production, which are seen as bubbles on top of the medium, and turbidity. It's pertinent to know that the bat alert blood culture bottles can also be analyzed without the bat alert system, and this can occur in scenarios where there are no constant power supply for 24 hours, and in that case, the bottles are incubated manually in the incubator at 36 degrees centigrade, plus or minus 1 degree centigrade, and are then examined daily for growth by visual check. With the reference to the image displayed, you can see that the visual check can be in the form of pellicle formation on the surface of the broth, gas production, which are in form of bubbles on top of the liquid part of the medium on top of it, and then turbidity. You could see that from the image. On the left hand side has no growth, while the right hand side, which is a turbid, indicates growth in the medium, and then puff balls at the bottom of the medium. For all manually incubated bat alert bottles, a subculture is performed after 24 hours and 48 hours. For the aerobic bottles, they are incubated on the blood and chocolate agar and incubated in a carbon dioxide enriched environment, while the anaerobic bottles are incubated on the chocolate agar and incubated in an anaerobic environment, and then a gram stain is performed accordingly. It's better to know that when other media can be used during the subculture, like the Maconche, with the target organism of the Enterobacteriaceae family, the non-fermenters, and the Pseudomonas species. For the sabored agars, the target organisms are the yeast cells and fungi. And also to know that for some special fastidious gram-positive cocci like the streps, most times it might require the addition of discs such as the optochin or bacitracin disc for ID of some of those organisms. The gram staining technique. Gram staining is a differential staining procedure which is used in the lab to categorize bacteria based on the composition of their cell walls. The principle is based on the ability of certain organisms to retain the primary stain even after decolorization. And for those that retain the primary stain even after decolorization, they are categorized as the gram-positive organisms, while those that retain the counter stain after decolorization are referred to as the gram-negative organisms. For this procedure to be carried out, certain supplies and reagents are needed, and these include, but not limited to, personal protective equipment, PPE, such as the protective coats, and then hand glove, the slide rack, timer, absorbent paper, water source such as the tap water or the ionized water, crystal violet, which is the primary stain, Logos iodine, which is the mordant, which takes the role of a mordant, the colorizer, of which acetone is preferably used. Then for the counter stain could be either safrani, neutral red, or dilute carbon fuchsin. And of course a bright field microscope with ×100 objective and an immersion oil. The gram staining procedure begins by making the smear from the blood culture bottles during subculture, and this is fixed by flaming on the Bunsen flame. After that, they are placed on the staining rack and flooded with the primary stain, which is crystal violet, for about 1 minute. After that, it's rinsed and flooded with the Logos iodine for 1 minute or so. This is then rinsed, and then the decolorization process follows. It is pertinent to know that at this stage, the timing is drastically reduced to prevent over decolorization. And this is done for 2 seconds, not more than 5 seconds. And it is washed off and then flooded with the counter stain for about 30 seconds. All in all, this procedure should not take more than 5 minutes. After rinsing the counter stain, the back of the slide is blotted dry and allowed to air dry before being examined under the microscope with the ×100 objective. At this stage, it's pertinent to note that the decolorization process could result in false positive, and as such, to quality control this procedure, it is pertinent to always stain known gram positive and known gram negative organisms alongside each batch of staining technique to quality control this whole procedure, and this will serve as a guide to ascertain whether the slide has been over decolorized or not. And also, in most cases, fresh isolate should be used as control. The image display makes reference to the final result, and for gram positive rod, they come out with a purple color, while the gram negative come out with a pink coloration. This is you can see different pictorial representation of the gram positive and the gram negative reaction. So it's also pertinent to know that for the gram positive organisms, there are different morphology in shape with terms to the shapes and arrangement. And for the cocci, you have the single cocci, you have the diplococci with 2 gram positive cocci fused together. You also have the ones in chains, such as the streptococcus. Then you have the ones in clusters, such as the staph, and then you have the tetrads, cocci in pairs of 4. For the bacilli, you have the single bacilli, you have the coccobacilli, you have the diplobacilli, and the curve-shaped gram positive cocci, and then the palisades. For the rods, you have the single rods or bacillus, you have the coccobacilli, you have the diplobacilli, the curved bacilli, and the palisades. And it's pertinent to know that most organisms in the gram-positive rods or bacilli category are usually contaminants, except they are otherwise clinically significant after careful review. Now, after all the gram staining and culture, it is very important to differentiate the pathogens from the contaminants. And this has to do with laid down guidelines using already established protocols in like the systematic bacteriology and then the CLSR guideline. It is also pertinent to know that any organism cultured from sterile sites, such as blood, CSF, or deep tissues, are usually considered of clinical significance, with few exceptions, such as skin organisms, such as coagulase negative staphylococcus species, grown in culture from a patient with no clinical sign of infection. But however, from superficial colonized sites, potentially pathogenic organisms need to be identified from mixed cultures and excluded from normal microbial flora. And for blood cultures, there are different categories of isolates that are of clinical significance, and this includes the enterobacteriaceae such as Klebsiella pneumonia, Escherichia coli, Salmonella species such as the typhi, Streptococcus species, Candida species. So, for the pathogen identification based on known guidelines and SOPs, there are certain pathogens that are of very clinical significance, and no matter any growth should be considered, and most of these organisms include enterobacteriaceae such as the Klebsiella pneumonia, E. coli, Salmonella species, Pseudomonas species, Enterococcus species, strep pneumonia, and other streptococcus species such as the Pyogenes and Agalati, and a whole lot more as displayed on the system. And for other classes of organisms, such as the alpha hemolytic streptococci, not including strep pneumonia, Acinetobacter species, Burkholderia species, and other yeast, other fungi, it is you can actually confirm that they are of clinical significance by having a repeat culture, and a repeat positive blood culture shows that they are of clinical significance for that patient. Organisms usually considered as contaminants include coagulase negative Staphylococcus species, diphtheroids, and Micrococcus species. In identifying pathogens and different types of pathogens, as said before, following the laid down guidelines is very important. And this includes doing a series of biochemical tests using specialized procedures such as the API's automated profile index, or using automated procedures such as the Vitek 2, MADI12, or Phoenix for identifying these samples. And all these procedures, followed by sensitivity testing, antimicrobial sensitivity testing, will go a long way in improving the health of these patients. Thank you.